Hi, my name is Roberto Serrano Pomales. I am a junior studying biomedical engineering at Brown University in Providence, Rhode Island. During the REU PR Climb program, I had the opportunity to collaborate on a project headed by my co-mentor Luis Bermudez in Dr. Eduardo Nicolao's labs. We identified how textile wastewater contains synthetic dyes that can be harmful to human health. A process called forward osmosis can help eliminate these pollutants via membrane filtration, but the microscopic pores in these membranes can get clogged by bacteria and other organic compounds, reducing its efficiency. The picture-perfect solution we aimed for in this study was to design and test a special membrane material to prevent these clots of bacteria, aka biofilm, from forming and to degrade the dye. The membrane is hydrophilic and water-attracting and would photocatalytically degrade the dyes using a photocatalyst tungsten disulfide to simultaneously kill the bacteria and break down harmful dyes via reactive oxidative species when the membrane was exposed to light. In the end, our study showed promise in degrading one of the dyes and preventing bacterial growth on the membrane surface, getting closer to our picture-perfect solution. See ya.
Hi, my name is Stephen Schnell, and I'm a current first-year undergraduate student at the University of Georgia, studying in the Department of Biochemistry and Molecular Biology. Last summer, I had the privilege of working under Dr. Cornelis Vlar in his medicinal chemistry lab. The picture-perfect solution that we worked towards was synthesizing novel drugs to treat malaria in an effort to overcome the anti-malarial resistance observed in nearly all strains of the disease today. As part of the project, I performed chemical reactions, isolated and purified the compounds that we synthesized using column and flash chromatography, and characterized our products using NMR spectroscopy.
My name is Liam Faubert. I am a senior undergraduate studying chemistry at the University of the Incarnate Word. And this past summer, I had the great opportunity of taking part in an REU program titled UPR PR Climb. In this program, I worked on a project developing anti-HIV drug derivatives, specifically on a class called maturation inhibitors. Maturation inhibitors work by binding to the HIV virus and blocking it from reaching the final form, which is in a spherical shape. When the virus is in a spherical shape, it is then able to infect other cells in the body. A picture-perfect solution to this project would be a drug that blocks this maturation process efficiently and consistently.
My name is Isha. I'm an undergraduate student studying chemistry and math at Rutgers University. As part of the 2024 REU PR Climb program, I worked in Dr. Noko's lab studying how metals can be used to develop innovative strategies against cancer cells. My project focused on two specific metals, titanium-4 and platinum-2. The purpose of studying titanium was to see how it binds to an iron-dependent enzyme called ribonucleotide reductase, which the cancer cells need for DNA repair and growth, and understand how this interaction could disrupt the enzyme's function. I also synthesized a platinum-triapene complex and tested its cytotoxic effects against cancerous and non-cancerous cells. And so the picture-perfect goal of this project was to contribute to the development of new and also improved metal-based cancer therapeutics by leveraging these metals to target and inhibit cancer cell survival.
Hello, my name is Gianmarco Feliciano Crespo. I'm a student at the University of Puerto Rico in Aguadilla. I'm part of the Natural Sciences Department and I'm currently majoring in biology. Let's get down into what we were doing. Mutations in the transcription factor known as TBX5 have been associated with congenital heart disease. And what we were trying to do is better understand that association. To do that, we were trying to look into how the functions of TBX5 were altered by these mutations. Now, this involved doing a variety of different processes, from understanding the thermal stability, if it can handle the heat without falling apart, its binding affinity. So a transcription factor can find that gene because it has an attraction to it, and that attraction is known as affinity. Now, we were studying how that affinity was altered. We were also trying to look into specificity. We had this gel, and we would combine TBX5, our transcription factor, with this massive library of DNA, and they were all different sequences of DNA. So remember I said that transcription factors are really picky over what they bind to. So by mixing together, it'll bind to its favorite sequences and it'll ignore the rest. And then we would run an electric current that would drag all the DNA through the gel. And as they were running through the gel, if TBX5 binded to that, it would weigh on it. We can compare the mutated variants of TBX5 to the wild type, or the healthy variety, and see how their specificity compares. And with that, we could try to find out if it is different, where is it binding to? And if it's binding to somewhere else, what effect might that have on the body? Now, the goal of this whole experiment is to open the way for more research that can hopefully lead to the development of cures or treatment options for congenital heart disease. Our picture-perfect solution was a large amount of information detailing how this mutated variant of TBX5 has been altered so we can better understand the mutation.
Hi, my name is Patricia Fontanes Carrion, and I'm a chemistry student in the University of Puerto Rico at Calle. This summer, I participated in the REU PR Climb program, where I developed electrocatalytic materials, specifically perovskite oxides, to enhance the oxygen evolution reaction in green hydrogen production. Now that might sound a bit technical, but it's all about finding better ways to produce cleaner energy. Our world needs more renewable sources to combat climate change, and hydrogen is a very promising fuel. By improving how we generate hydrogen, we can also reduce our dependency on fossil fuels. So it's like working towards a picture-perfect future where energy is accessible, cleaner, and abundant. And that's what chemistry can do.